Hi, this is a very introductory lecture about bacteria. If you go on to take microbiology, you will learn a considerable amount about bacteria. This is just meant to review what a prokaryotic cell looks like, the way it divides, some basic characteristics of bacteria, and then looking at some beneficial roles of bacteria in addition to a short list of pathogenic bacteria. So just a reminder that bacteria are prokaryotes. And remember that when we're talking about a prokaryote, we mean that the DNA is not enclosed in a nuclear envelope. That is the primary characteristic defining this cell as a prokaryote. Remember, pro means before, and karyote is referring to the nucleus, so before nucleus. So that DNA is just free in the cell. In addition to what we call the chromosomal DNA, which is right here, we also have this other DNA called the plasmid DNA. The plasmid DNA differs from the chromosomal DNA in that it codes for non-essential proteins. The chromosomal DNA codes for all the proteins required for this bacterial cell to carry out its basic function, its basic metabolism. Remember that prokaryotes do not have any membranous organelles, so they do have ribosomes for protein translation. Those ribosomes are considerably smaller than eukaryotic ribosomes. In addition to that, we're going to just quickly look at the structures surrounding the cell. So remember that all prokaryotes are single-celled organisms, meaning the entire organism is just one cell. There are no multicellular prokaryotes. Surrounding the cell is a plasma membrane that is made of a phospholipid bilayer, just like all membrane. And then outside of that is a cell wall. Now this cell wall is shown in yellow here. It is different in structure from the cell walls we've seen in other organisms. We've seen a cell wall in plants that was made of cellulose, which is a chain of glucose. And there was a cell wall in fungi made of chitin. This is made of something called peptidoglycan. And I'm just going to skip ahead really fast to a future slide so I can show you the word peptidoglycan. So here's peptidoglycan. The peptido part, that is protein, and then the glycan, that's carbohydrate. So it's a combination of protein and carbohydrate that's making up the cell wall of that bacteria. Okay, and then some bacteria have a capsule on the outside of that cell wall. That capsule makes those bacteria who have it very resilient to environmental extremes such as UV radiation, heat, desiccation. These pili, shown here, will allow these bacteria to stick to surfaces, stick to your intestinal wall. And finally, some bacteria actually move. So this would be a bacterial flagellum that would allow this cell to actually move. Bacteria have asexual reproduction, and the specific mode of asexual reproduction they use is called binary fission. They basically copy their DNA. So you can see they started with just this black piece of circular DNA. It's copied, moves to two ends of the cell, and then new cell wall and plasma membrane are laid down until we have two separate identical cells. Binary fission. This happens very quickly. So bacteria can reproduce very simply and quickly, becoming millions in a very short period of time. The way we identify bacteria, you will learn a lot about if you go on to take microbiology. This is just a quick list of some basic ways. So shape, where they are found in the body or in the environment, whether they're gram positive or negative using a test called a gram stain, their oxygen requirements, whether they have a capsule or not, whether they grow on certain types of media or not, and other tests that can be performed. These are the three basic shapes of bacteria. Usually when we draw a bacterial cell on the board, we draw this rod-shaped bacterium called the bacilli. These are cocci. If you've ever heard streptococcus or staphylococcus, it's referring to these spherical bacteria. This has two stuck together, so this would be called diplococcus. And then finally, spiral would be the third category of bacterial shape. Then if you pair that with where they're found, so say they're rod-shaped bacteria and they're found on the skin, or they're spherical and they're found in the throat, that can narrow down to a smaller list just based on those two characteristics. One of the most basic skills, not basic skills, one of the most basic tests you will learn to do if you take microbiology is a gram stain. And the gram stain is really staining bacteria that have a very specific configuration of their cell wall. So these bacteria on the left have this thick outer peptidoglycan layer right here. So they have this external cell wall. You can see it all the way around. It looks like a tic tac. But this thick peptidoglycan layer on the outside. And if a purple stain called crystal violet is applied, they stain purple. And you can see them here. We call them gram positive because they do stain when you go through this gram staining process with this dye. So you can see the dark purple ones here. So let's say you prepared this slide and you stained it according to the procedure, which I won't go into the details of, and nothing was purple. You wouldn't know if you had just rinsed off all of your bacteria or if they were all gram negative. So we apply something called a counter stain that stains all bacteria, and it's pink to red in color. And if you look at these bacteria that are called gram negative, they have stained pink. The pink stain has stained these purple ones too, but it just doesn't show up over the purple. So these would be gram negative, the ones that stain pink. And the reason they don't stain purple is their peptidoglycan layer is actually between the plasma membrane and an outer membrane. So it's sandwiched in between here. So these gram negative bacteria do not stain purple with crystal violet. So that tells us something about the bacteria as well. So this is a bigger picture of that. You can see the gram positive ones are purple, the gram negative are pink or pink to red in color. And we would refer to these as gram positive rods. This would be gram negative rods, gram positive cocci. Nutritional modes of the bacteria can tell us something about what we're looking at. So photosynthetic bacteria are called photoautotrophs. Remember, autotroph means they can make their own food. There are also chemoautotrophs who can make their own food from different chemicals rather than from light. So photo is referring to light. Heterotrophs, of course, eat their food. So most bacteria are heterotrophs. Oxygen requirements in bacteria vary. So some bacteria require oxygen. And remember, aerobic means with oxygen. And obligate means you're obligated to this lifestyle. So if you're an obligate aerobe, it means you must have oxygen to survive. So these bacteria require oxygen versus obligate anaerobes. Remember, anaerobic means without oxygen. So they're obligated to have a life without oxygen. In fact, they are killed by oxygen. Okay, and then finally, facultative means you're not obligated to do that, okay? Means you have options, but there are only facultative anaerobes. There are not facultative aerobes. So facultative anaerobes, they don't require oxygen, but they're not killed by oxygen either. So the example given is E. coli. You probably know these live in your gut, in your colon, and so they can come outside the body and be infectious to other people. So they're not killed by oxygen, but they do well when oxygen is not available. This would be a picture of a bacterial cell surrounded by a capsule. So that would be another way we could identify what we're looking at is if we happen to see a capsule. And then different media for growing the bacteria. You can see that these two different species of bacteria reacted differently on this mannitol salt agar. So the Staphylococcus aureus grew and these other bacteria did not. We can look at whether they have respiratory membranes, whether they have thylakoid membranes, making them photosynthetic. Remember, the thylakoid membranes are where photosynthesis takes place. Remember, this is very similar to mitochondria within eukaryotic cells. This would be a picture of cyanobacteria, which you learned in photosynthesis carries out photosynthesis. So some bacteria are photosynthetic. And then this is just looking at the chromosomal DNA versus the plasmid DNA. So the plasmid DNA are these small circular pieces of DNA code for non-essential proteins versus the chromosomal DNA is one big circular piece of DNA and it's just folded in the cell, not enclosed in a nuclear envelope, a hallmark characteristic of prokaryotic cells. I'm going to skip that one. Okay, this is a very important process to understand. Bacterial conjugation. It really means bacterial sex. Certain bacteria have a gene that allows them to grow this structure called a sex pilus. It can also be called a conjugation tube. And what this allows them to do is they can transmit plasmid DNA to another bacterial cell. So let's say that this guy has a piece of plasmid DNA right here, and that it happens to have a gene on it for antibiotic resistance. And he also has a gene that allows for the transfer of that plasmid DNA to another cell. So it will make a copy of its plasmid, and it will then transmit that copy to this other cell, thereby transferring all the genes, including that gene for antibiotic resistance. And now a bacterial cell that was not previously resistant to a specific antibiotic can become antibiotic resistant. So that's kind of scary in a clinical setting that bacteria have the ability to do that. Here's another picture of that happening. So this bacterial cell has made a copy of its plasmid and it's going to transmit that copy to the other cell, and now that other cell has a copy of that same plasmid DNA. Bacteria get a bad rap, but most bacteria are actually beneficial, and this is just a short list of some of the many beneficial roles of bacteria in our environment. Okay, so decomposition. Not only does this mean we're not piled high in everything that has ever died since the beginning of time, but it also means that we're able to return nutrients to the soil. So we'll look at that briefly. Nitrogen fixation, which is incredibly important. Bioremediation means using biological organisms to remediate, clean up the environment. Making pharmaceutical products, carrying out viral research, making specific food products, digesting cellulose in the guts of certain animals, and then our own microbiome, our own symbiotic bacteria. Okay, nutrient recycling, incredibly important on Earth. We do not have an infinite amount of the critical nutrients that living organisms need, in particular things like nitrogen, phosphorus, sulfur. These have to be returned to the earth and taken up by new plants. And we count on bacteria to do that for us. Decomposers. Decomposers break down organic matter, and the soil stores those nutrients, and the plant then absorbs those nutrients and grows, and then other organisms eat this plant or eat someone who ate the plant. So decomposition is an incredibly important role of bacteria on Earth. Bacteria play an incredibly important role in the nitrogen cycle. Most nitrogen on Earth exists as nitrogen gas in the atmosphere. Plants and animals do not have the ability to take nitrogen gas into the body and use it to make molecules. Why is that a big deal? Well, let's think back to which molecules need nitrogen. Okay, the first one I'm going to remind you of. Oops, that's way too big, sorry. I'm just gonna draw it with a pen. Let me just do blue here. Okay, oops, sorry. I wish I had my iPad right now, but someone borrowed it. Okay. Carbon, hydrogen, carbon here. This is going to be a carboxyl group. Sorry, this is terrible writing. Doing the best I can with my mouse. Sorry about all the clicking. Do an R group here. Well, I'll just do a hydrogen. We'll make this glycine. Okay, this is an amino acid. Amino means nitrogen. So this amino group on the amino acid, all amino acids that make up our proteins require nitrogen. That's pretty dang important. Okay, so nitrogen right there. The other macromolecule that has nitrogen is nucleic acids. I should say are nucleic acids. The other macromolecules that require nitrogen are nucleic acids. So ATP, DNA, ADP, RNA, all require nitrogen. And if you'll recall, the basic structure is that you have a pentose sugar, ribose or deoxyribose. You have a phosphate group, or 2 or 3, depending on which nucleic acid we're talking about. And then this is called a nitrogenous base. It's called a nitrogenous base because part of the structure of this base, whether it's A, C, G, T, or U, it contains nitrogen. Okay, so nitrogen is necessary to make nucleic acids and to make proteins. Incredibly important. The problem is we can't use nitrogen gas. Who can use nitrogen gas? Certain bacteria who can do something called nitrogen fixation. Okay, these bacteria can take nitrogen from the atmosphere and do something called nitrogen fixation, where they convert that atmospheric nitrogen gas into a form that plants can use. And it can then be taken up by the roots of plants. And those nitrogen fixing bacteria can live on the roots of certain plants in these little nodules. These are symbiotic bacteria on the roots of this legume, or they can live in the soil. So that's an incredibly important role of bacteria on Earth. Here's a picture of some rhizobium, which are nitrogen fixing bacteria, and they're living in a nodule on the root of this legume. Bioremediation, environmental cleanup. There are bacteria who can eat almost anything: pesticides, oil, sewage, plastics, other toxins in the environment. In San Diego County, our major sewer treatment plant is down at Point Loma, and it uses primarily biofilters that contain bacteria that decontaminate the sewage, break it down to where it's no longer harmful, and that sewage is then released into the ocean without causing harm to the organisms because of those beneficial bacteria that clean up the sewage. We make pharmaceutical products using bacteria. So bacterial parts are used to make vaccinations against bacteria. There's an important process called bacterial transformation, in which you insert a gene into the bacterial genome and the bacteria will make that protein. So insulin for diabetics is made using bacteria. And we also make certain antibiotics using bacteria. Bacteria are huge in viral research. We know a lot about viruses based on research we've done, in particular with E. coli. Okay, much easier to do viral research on bacteria versus a mammal. The viruses that infect bacteria are called bacteriophages, and the short form of that is phages. So phages are viruses that infect bacteria. This is a list of just a few food products that we make using bacteria, all of these different dairy products, all of these fermented products, processing coffee and chocolate, curing meats, making soy sauce. So bacteria can be our friends when they help us make these yummy food products. If you're a mammal who has a high cellulose diet, remember that animals do not have the enzymes to digest cellulose, to break it down into glucose. And yet this cow is not going around eating a cheeseburger, right? Because that would be cannibalism. Okay, so this cow is eating grass and it can't digest the cellulose and break it into glucose to use for energy. Instead, they count on the symbiotic bacteria in their digestive system, in their guts, to digest that cellulose for them. Symbiotic bacteria. That's incredibly important. And then there are different bacteria in the rabbit as well. Okay, last category of beneficial bacteria are those that make up the human microbiome. These symbiotic bacteria that live on us and in us are 10 to 1 the abundance of our cells. So we have 10 bacterial cells for every 1 human cell. We are mostly bacteria, and we're learning more every day about what our microbiome does for us, how critical it is to our health and well-being. Some scientists are even saying it's more important than your own DNA in determining your health, both your mental health and your physical health. Depending on who your instructor is, you might have a separate video to watch on the human microbiome. This is just a short list of the beneficial roles of those symbiotic bacteria that live in you and on you. They dictate your mental health, your immune function. In fact, we're learning more and more about how bacteria train our immune system to do its job. Digestion of your food, absorption of nutrients, producing vitamins that we cannot get in our diet, things like vitamin K and certain B vitamins that are all manufactured by symbiotic bacteria in your colon. Tissue repair, whether you lose or gain weight, protection against certain pathogens, regulating blood sugar. These are all many of the important roles of bacteria in your gut and throughout your entire body. And there are many more than that. So this slide just kind of summarizes that. We have 10 times more microbial cells in our body than human cells. That's pretty crazy to think about. So extracting energy from our food, producing vitamins, regulating our immune system, regulating glucose levels, protecting us against disease. How does that happen? Well, if you have this whole community of good guys, they're out competing the bad guys for space. If you take an antibiotic that kills all of these good guys, guess what? The bad guys can move in. They're called opportunistic. And they can move in and take over when you kill all the good guys. Microbial communities shift throughout a person's life. They're influenced by diet, exercise, medications, and other environmental factors and pathogens that you're exposed to. Okay, so lots of beneficial roles of bacteria, but not all bacteria are good, as we know. So now let's just quickly talk about the pathogenic bacteria. Remember, pathogenic means disease-causing. If an organism is pathogenic, it means it causes disease. You will learn a lot about pathogenic bacteria if you take microbiology. If an organism is a strict pathogen, it means it causes disease in everyone, even a normal, healthy person. Versus an opportunistic pathogen usually only causes trouble if you're immunocompromised, if you have another illness, if you take a lot of antibiotics that kill the good guys and now the bad guys can move in. So those are called opportunistic pathogens. This is how bacteria get into the human body, usually through mucus or skin. When we say mucus, we mean your respiratory tract, your digestive tract, your urinary tract, your reproductive tract. Those are all ways that bacteria can get in, or through your skin. Routes of entry into these areas: ingestion, means you eat them; inhalation, you breathe them in; trauma, an injury; needles, catheters; an arthropod bite. So this means a spider, a scorpion, a tick, an insect; or sexual transmission. These are some different ways that bacteria can cause disease. They can destroy tissue. They can produce toxins, or they can cause an overwhelming immune response. When your body has a strong inflammatory response to something it perceives as foreign, that alone can kill you. So oftentimes your immune system's response to the foreign cell or particle, such as a virus, is going to be what kills you, not the pathogen itself. Here's a short list of diseases caused by bacteria. These would all be respiratory. Many of these we have vaccinations for now, but they're still pretty common. The skin, intestinal bacteria, and reproductive bacteria. These reproductive diseases are increasing in frequency as it becomes easier for people to hook up through online dating. And so we're seeing diseases that were mostly eradicated in the United States start to peak again. These diseases used to really kill a lot of people on Earth. And in fact, if you look through history, you can see a lot of famous historical figures who had syphilis and even gonorrhea and suffered from symptoms of those for their entire lives. Okay, very important to realize that antibiotics only kill bacteria. Okay, so let's look at this word. Anti means against, biotic means living. It has to be a living organism to be killed by something called an antibiotic. But more specifically, it's bacteria that we kill with antibiotics. We do not kill viruses with antibiotics. Antibiotics do nothing to a virus. When you have the flu, antibiotics will not help you. How do antibiotics work? Well, they're going to target different processes in the bacterial cell to either keep it from reproducing or keep it from functioning, or both. Okay, so keeping it from making new cell wall is going to keep it from dividing, keeping it from making proteins, interfering with its production of nucleic acid so it can't copy its DNA, interfering with the cell membrane production, or any other type of metabolism in the antibiotic. Antibiotic resistance is a huge problem in medicine today because we have taken too many antibiotics and we don't have new ones being produced. Most of our antibiotics have been around for quite a while. I know that I was recently prescribed penicillin, which was our very first antibiotic. Believe me, so incredibly important in human history, increased human lifespan dramatically when we invented antibiotics. And people used to just die from things like a tooth infection, things that we just take for granted now that we can treat. So this is what happens. It's natural selection. Antibiotics are going to kill everyone who's not resistant, but we have just these two guys here, these two bacterial cells that are resistant, and those are the ones that survive and reproduce. And now we have this entire population that's resistant. What does the resistant means? It means they're not killed by standard antibiotics. How are they resistant? They just happen to have a gene that causes them to have one of these processes that's just slightly different. And our antibiotic is targeting a very specific part of this process, oftentimes an enzyme. And remember, shape is key. If something has a slightly different shape, then that drug isn't going to work. So DNA mutations offer a survival advantage for these bacterial cells. And over time, these become more abundant and harder to kill. And even taking this a step further, bacterial conjugation, if this guy can transfer antibiotic resistance to another cell, then that's a really bad thing. Why do we not develop new antibiotics then? Well, the problem is there is no financial incentive for the pharmaceutical industry to develop new antibiotics because the profit isn't there. If you're going to spend millions of dollars on preclinical research and clinical research, and then you only have a patent for a certain period of time, and you've put all this money in, and it's going to take on average 23 years to start gaining a profit on the antibiotic. So that's why we don't develop new antibiotics, even though we need to combat antibiotic resistance. Okay, that's it for bacteria. Thank you.